Okay, let's get started. Wouldn't it be funny if I just laughed? Okay, here we are. Quick business. Quiz 1, some tips for success. I got a few panicked emails. Every semester starts with a few panicked emails because it takes time to figure out how to take my quizzes. I'm sorry for the learning curve. You'll be okay, I promise. The average was like a 70-something on the quiz, so that means it wasn't a disaster. That means we're okay. Don't forget, you keep your 10 best quizzes. There's room for error. I think we'll have like 13 or 14. So it's going to be okay that there's a learning curve. This won't hurt your grade. You can be mad at me. It's okay. So tips for success. 1, do not just read randomly and then close your book and put it away and try to take a quiz. Really have your book out in front of you. Use Google, use NCBI books, use everything. People are saying that what I was quizzing them on was not in the book. I go straight from the book. I promise you it's in there. But even so, who cares? Find it. You can find it. I promise you can do it. It's just really about figuring out how to find things. The other thing is don't just scan and look for answers. It's not going to work that way. So sometimes you might need to read about a whole topic, then go back, look at the question, think about it, look at all the answers. Maybe a few of the answers will make sense to you. Look those things up. It's going to take a little time. It's not something you could just like bang out in an hour or 10 minutes, maybe an hour. I don't know. But one student emailed me and said, can you please put a list of topics so at least we have something to go with? Because there's no chapters, there's no pages for reading. And I'm kind of like, well, you have to just figure it out. But then I'm like, well, a list of topics might be helpful. So I'm going to try with the announcements to at least put major topics. Like, so, you get an announcement in on Blackboard when the quiz is posted. In that announcement, it'll say, this quiz will cover signal transduction, receptors, like these certain topics, which hopefully then you can start to direct your reading a little bit better. Let me know if it works. I'm always open to the advice of students. The thing is, if you're going to email me to complain, have a suggestion, and maybe we can make things better. Okay, don't forget to take the pre-test, due on Friday. I already noticed a lot of people forgot yesterday's quiz. I think about 15 of you. So, not good. Don't forget to bring Scantrons to recitation, because that's for the recitation quiz. You're going to need to have that. And then there's one Scantron you'll need for the final. Please bring that to me. Don't ask me the numbers. It's in the syllabus. Just take the syllabus to the bookstore and match up your numbers. It's all good. Okay, that's business. Okay, so we're going to start with writing. Because, well, we're trying something new. It's fun to do new things. So I want you to take the next 5 minutes to write. And this is what you're going to write about. This secretary pathway. You read about it this weekend. Take the quiz. And don't just answer these questions. It's just, I want you to kind of start to get an idea of the depth that I want in my questions. So what is it? Why does it exist? How do proteins enter? What is the path for proteins in this pathway? It goes from here to here to here. What happens in each component of the pathway? Do the best you can. The goal is that when you finish this unit, you can go back and look at this and go, oh, now I really get this. Hopefully. Okay. So how was that? Do you feel like, well, we'll see. Okay. So I think as we get further into the semester, we'll start going over. The questions will become more specific and more related to what we learned in the previous lecture, but this, there was not much of a previous lecture. And we'll start to go over what we wrote. But you'll notice the questions are really kind of open and vague and big. And I think it's because I really want you guys to be thinking about the big picture, not these little details. So if you can get through my whole class, I want detail. You can ask anyone who's had me. I like detail. But if you don't remember the big picture, it'll be hard to figure out where to put the details. Okay, so let's start. Unit 1, intercellular traffic. And so that's really, everyone's got rustling of papers and whatnot. That's just really about proteins going where they need to go to do what they need to do. And the textbook, I'm pretty sure the most recent version has this too. The textbook has this nice kind of map in the beginning of each section of this topic, and I like that because it kind of shows you the path that proteins can take, right? So you can go from the cytosol, as in a ribosome beginning to translate a protein in the cytosol, to the ER, and that's what we're starting as we talk about the secretory pathway, right? So that's cytosol, ER, Golgi, and then you can go anywhere from Golgi to the endosome lysosome, to the cell exterior, to the secretion, and then also membrane proteins. But then also remember you've got proteins that need to go to the nucleus, proteins that need to go to the mitochondria, and so on. And so it's kind of useful to kind of think of it as this map in terms of the different places that proteins can go. Why do they need to go to those places? Because proteins do things. And they need to do things in the places that they're meant to do things. Okay, so I want you to take a minute to look at this picture, because we're going to start getting into the nitty-gritty of this protein does this and this protein does that. But I want you to think about the big picture, which is that you've got your nucleus, and in your nucleus you've got your DNA, and you get transcription, and then you have your RNA that comes out and it needs to be translated, right? And so there are a lot of places for proteins to go, just like the map. Some people like that map, some people like the picture, right? So you're going to have your RNA coming out of your nuclear pore. It's going to associate with ribosomes, right? And you're just going to get translation. And you can get free ribosomes translating in the cytosol. You can get ribosomes that end up being dragged over to the ER, and that's our secretory pathway, and then they go ER, Golgi, and then out. You can get, then you'll also have, of these proteins being translated in the cytosol, there will be targeting sequences that will take them to the mitochondria, to chloroplasts, to peroxisomes, back into the nucleus. So just remember, all of this is happening. It's all happening at the same time. So if you were to go inside the cell, you'd see proteins zooming around all over the place because they need to do what they need to do to keep the cell going. Okay, so just whenever you find yourself deep in these details and you don't know where to put the details, go back to this picture and be like, okay, what are we talking about? And hopefully that should help orient you. Okay, so the first stop in the secretory pathway is the ER. So the main points here, proteins need to go where they work. And the secretory pathway is for proteins that are ultimately secreted, membrane proteins, and resident lysosome proteins. The other thing to keep in mind is there are proteins that work in the ER and Golgi, and they need to get there too. So they're also going to enter the secretory pathway. They're just going to be retained at certain points in the pathway. And we will learn about that. So we're going to talk about how we study the secretory pathway, because all of this stuff we're learning, this is someone's research at some point, right? So there are methods for studying the secretory pathway. We're going to talk about how proteins get into the secretory pathway. And then we're going to end with talking about membrane proteins and how they originate in that ER membrane. okay, and how that works. Okay, so how does a protein get into the ER? Who can tell me? You guys all just spent 5 minutes writing, so you're on it. How does a protein get into the ER? If people don't raise their hands, I will call people. Yeah. Right, so it's translated into a pore into the ER, right? Right. Does anyone want to add to that? Yep.
It's binded by a signal receptor protein.
Okay, so SRP. SRP is signal recognition particle, but it acts the same. You got it though. SRP signal recognition. And I had to think for a minute. I'm like, but that makes total sense. These words make sense. Signal recognition particle, right? So it's bound. So there's this. What's bound by the SRP? Signal sequence, right? So signal sequence is recognized by SRP and drags the whole thing over to this pore that then translation is going to resume. Anyone else want to add to that? That's pretty much the gist of it. I mean, we'll go into it in more detail, but that's the gist of it. Okay, so there's two ribosome cycles. You've got your free cytosolic ribosomes that are translating proteins that are either cytosolic or they're going to be imported into other organelles or the nucleus or something or other. And then what we're focusing on right now are these ER-bound ribosomes. And these are these ribosomes that are bringing the proteins into the secretory pathway. And so that's down here at the bottom. And this is mediated by SRP recognizing a signal sequence. Right, so you can get delivery of protein to the ER via post-translational or a co-translational mechanism. Post-translational, that means after translation. So that's the fully translated protein can be brought into the ER. But more frequently, and what we hear about the most, what we think about when we think of rough ER is co-translational import. And that's what we're going to talk the most about here. Co-translational at the same time, right? So the protein's being imported into the ER during a translation. So you've got your mRNA, your ribosome, your protein, and then this red part here is your signal sequence. And that's what's going to be recognized by your SRP. And then SRP binds to a SRP receptor on the ER membrane and brings the whole thing over. And then translation occurs right through the translocator. So what happens is you've got a ribosome in the cytosol, and it's translating, and the first little bit of the polypeptide that comes out will have this signal sequence. It's a specific amino acid sequence that is recognized by SRP. And that's how it's targeted to the ER. And then you'll notice right here it says cleavage site of signal peptidase. Well, there's a protein in the ER called signal peptidase that will cleave off the signal sequence. So you would think that a protein outside that's translated outside the ER, if possible, versus inside the ER will be different sizes because you're going to get cleavage of that signal sequence. Okay, any questions so far? I'm going to start nice and easy. Okay, so the SRP is going to bind to the signal sequence, and then it's pausing translation, and it brings the whole complex over to the ER. And so this right here, this brown blob, that's the SRP binding over there to the signal sequence. And then it actually has a subunit on it, not a subunit, but a part of the protein that stops translation. So it pauses it, brings it over to the ER. And this week, we're going to be reading a paper, right, about SRP, about the identification of SRP in recitation. Hopefully, half of you read it, right? Already? Done. Okay, so your signal sequence is recognized by SRP, and it's going to recruit the whole complex to the ER through binding the SRP receptor. So here, sometimes I'll show you multiple figures showing the same thing because one figure might be easier to see than another. So this is just another way of looking at it. You've got your mRNA bound to your ribosome, and you've got your translation beginning, and the signal sequence is coming out. And SRP is going to bind to it, bring the whole thing over, binds to SRP receptor, which is bound to the ER membrane, right? That's how it's going to get there. And then you're going to get translation. And then inside the ER, this round thing here is the ER. Inside the ER, you've got your signal peptidase, and it's going to cleave off your signal sequence. So again, the whole ribosome mRNA polypeptide complex is brought to the ER membrane through binding of the SRP to SRP receptor. Okay, so then you have to think to yourself, okay, so now what? Yeah, so it's got to go through, it's not going to go through the ER membrane, right? Can a protein go straight through a membrane? No, that makes no sense at all. Right, so there's a pore multiple pores in the ER membrane that are called translocators, or I might sometimes call them translocons. So what does translocate mean to you? To cross something, right? So I want you to always, in biology, stop and think about the word, because usually what it does is within the word, right? So this is a translocator, so it's involved in translocating the protein across that ER membrane. And so we can get very complicated and call it the Sec61 translocator. But even if you just remember translocator or translocon, that's good for me. So this is the structure of the Sec61 translocator. And it sits in the ER membrane, and it can be closed. So it's actually got a protein structure within it, a polypeptide that acts as a plug, or it could be open. And so this is kind of as if you're like standing inside the ER looking up through the translocator, right? So here, this green part would be your protein coming through, and remember we saw that signal sequence attached to the side, like over here and there. So that's what you would see. And then the other thing I want you to remember here is that we're going to talk about membrane proteins and how they are translated directly into the ER membrane, and how then once they hit this stop transfer sequence, which is essentially this hydrophobic stretch of amino acids, then they're going to move into the membrane right then and there. And so you can see how that kind of might work, because they call it the seam, where two different chains of the translocon come together. It actually can open. And you can see in this figure, the signal peptide is actually sitting within that opening. So if I were to draw the ER membrane, it would be kind of coming off this way, right? So you can imagine that this could move right in, and you would have an integral membrane protein. I always lose people when I start talking about membrane proteins. So I want you guys to, I don't know why, I have no idea why, but I want you guys to stop me right away as soon as you start getting lost, if you get lost. But I've tried to trim that part of the lecture, so hopefully we won't get lost this year. Okay, any questions? I'm doing a lot of talking. Okay, so the ribosome is binding to the translocon, and it translates the protein through the translocon into the ER. Okay, so this is talking about co-translational import. Then there was this post-translational import that I spoke about. You can kind of compare the two. This is what we've been talking about, where you've got your, it's actually the translation that's driving the translocation of your protein into the ER. And in post-translational translocation, you already have a fully translated protein, and it needs to get into the ER, or in the case of bacteria, into the extracellular space because is there an ER in bacteria? But it's conserved machinery. So here you actually need some kind of energy. So in eukaryotes is in the form of BIP, which is an HSP70 family chaperone, and it uses ATP to kind of use this ratcheting motion to pull down the protein through the translocon. And then here, this Sec-A has ATPase activity. So it's also using ATP to kind of feed, you can imagine feeding thread through a needle, right? It's kind of feeding the protein through the translocon. So if you guys had compare and contrast, really the difference between these two is the force of the protein going through is really due to translation, and the other is the ATP, that's the ATP-dependent process of feeding the protein through the translocon. Yep, so in post-translational translocation in eukaryotic cells, chaperones in the ER lumen within the ER are helping to kind of pull the protein through. So it's this kind of ratcheting motion of the chaperones, and that's ATP dependent. Any other questions? Who's having fun? Yeah, that was perfect. Okay, who's gonna have? I want all of you to change your ringtones to a laugh track. Like maybe half do laugh track. No, okay. A third do laugh track. A third do booing, because sometimes there will be need for booing, and then a third can do like a woo kind of. Yes, laugh track. What is an integral membrane protein? It's just a membrane protein. It's integral because it goes through the membrane, so it's got this about 20 to 30 amino acids in its sequence are hydrophobic, and they create that transmembrane domain. And so the reason I bring it up here is because membrane proteins originate in the ER and go through the secretory pathway. That's how they get to the membrane. Okay? Okay. Laugh track, booing, wooing. Oh, I had this dream last night. I was going to tell you guys about it. I was just thinking to myself, and you know, you wake up and you forget your dream, but then like you randomly remember it mid-morning sometimes. I had this dream that there was this big play, like I'm talking like Broadway size big play that was happening, and one of the actresses couldn't be in it, and they asked me to fill in, but I had never even seen the play. I'd seen it like half of it once. And so I was doing what I thought I should be doing, and I was doing a horrible job. And they actually pulled me off stage. Not with a cane, but they just, I don't know, somehow asked me to go off stage and not go back on. And I was like, well, this is so unfair. You guys didn't even tell me what the play was about, and now you're firing me. And I thought to myself, I wonder if I'm nervous about teaching. That's weird, because I've taught this class a few times. Maybe I don't know, guys. Might be a little nervous. I don't know. Anywho, weird dream. It was so realistic. That's where like these weird foot scrubbing thingies. I don't know. Okay. Co-translation, post-translation. Okay, so once you're in the cell, signal peptidase cleaves a signal sequence inside the ER, and that releases the protein into the ER. So as you notice, you have the protein kind of being fed through as it's being translated, but the whole time your signal sequence is bound to your translocon, and so it's going to get longer and longer, and then eventually it's going to cleave, and the whole thing will be released into the ER. Okay? Okay. Uh-oh. Okay. So you might ask, well, Dr. Ehrlich, how did they learn all these things about import of proteins into the ER? Tell me. So I'm going to tell you a little bit of something. Basically, what you can do is you can make these microsomes. All you have to do is take a cell and You can isolate the ER, and then you can disrupt it. And remember, the ER is made of membrane, and so what happens to little pieces of membrane in solution? They kind of turn into a little ball of membrane. Can you guys imagine that? Because they don't want to, like, they're not just going to, like, stay there with their hydrophobic parts being all exposed. They want to kind of close up, right? So only those polar heads are exposed to the surrounding buffer. So by disrupting the ER, you're going to get these little spheres of ER that are called microsomes. And you could have rough microsomes or smooth microsomes because you have rough or smooth ER, right? And so then you can centrifuge them through a gradient of some sort of dense media. We won't worry about that now, exactly how that works, but really what you're doing is separating things based on density, right? And your smooth microsomes and your rough microsomes, are they going to be at the same place after you centrifuge them? No, they're going to, because they're going to have different densities, so they're going to band at different places, and that's what's happening here. Your smooth microsomes, are they going to be lighter or heavier than your rough microsomes? Lighter, right, because they don't have ribosomes. Right, so when you centrifuge, things are going to move down, right? They're going to pellet based on their size, shape, weight, density. And your rough microsomes are going to be closer to the bottom. So you can isolate them, and then you can do experiments with them. So this is really just about isolating them. There's no science here yet. No science at all. Just the method. So,
I didn't know that the microsomes will be all the same size, like when you do cell destruction.
how do you know that the microsomes will be all one size when you do cell disruption? Okay, so that is a really good question. And I think it's really dependent upon how you disrupt. So normally, I believe you would sonicate the cells. But either way, so let's say you disrupt. If you disrupt for a uniform period of time, you would expect that you would have equal amounts of heavy and not heavy, of rough and smooth. And they're not all going to be in one thin band, they're going to be in a range. Because they will vary a teeny tiny bit, but you can definitely separate your smooth and your rough because that difference is going to be much greater. Does that make sense? But what you would probably do, let's say you were working in the lab, is you would do a trial where you would be like, I'm going to disrupt for 30 seconds, for 1 minute, for 1.5 minutes. I'm going to look, I'm going to spin them down, I'm going to look and see what gives me the best concentration of my rough microsomes. Okay? Any other questions? That was a good question. I like it when people are thinking about methods. Okay, so now that we've done that, why am I telling you about this? Well, this is how we study things, right? So, again, once you disrupt and then you're going to put it on your gradient, here it's a sucrose gradient, which really just means that it's, you guys know what sucrose is, right? So you'll have a gradient of, an increasing gradient of sucrose, and you'll have your microsomes on top, and as you centrifuge, they're going to move through until they reach their equilibrium. And then you'll isolate your smooth or your rough. Okay, so now you've got your microsomes. What are you going to do with them? Well, you could study things, okay? First, I want you to think about why they're useful for studying. So microsomes are basically little ERs. They've got the same topology and function as the ER. Topology, what does topology mean? I'm going to use that a little bit over the next few lectures. Does anyone want to take a stab at what topology might mean? I guess. You know, typically the same. Think more about your what is where on your microsomes. Same structure. So you're, yeah, yep. Were you gonna raise your hand? The study of layers. Okay, so you're, yeah. So when you look at your microsome, what's on the inside remains on the inside, and what's on the outside remains on the outside. So in your ER, where are your ribosomes, on the inside or outside? Outside. So when you disrupt and you get your microsomes, your ribosomes will still be on the outside. If you've got a protein that was in the middle of being translated and it's a membrane protein, then if the N-terminus was on the inside before, it will remain on the inside. If the C-terminus of that protein was on the outside before, it will remain on the outside. So maintain topology. And we talk about that a little as we get into membrane proteins. So if you've got your microsomes with your ribosomes attached, and you treat them with detergent, what does detergent do? It solubilizes membrane, right? So now you destroy your microsome. And if you add protease, what does protease do? It degrades proteins, right? And so if you treat with detergent and then you treat with protease, will you have any protein left in your microsome? No, because you destroyed it all. But if you don't treat with detergent, you only add protease, will your protein billet still be present? Yes, because you have that microsome that is covering and protecting the protein from being degraded. And so it's its own continuous membrane with protein, whatever secretory protein that's being studied on the inside. Okay? So microsomes were used to study the signal sequence hypothesis. And so we've already talked about signal sequence, but there was a time that it wasn't in a textbook, that it was a hypothesis. People hypothesized, well, maybe there's this signal sequence, this bit of protein that directs secretory proteins to the ER, and then it's cleaved off inside the microsome, inside the ER. Maybe there's an enzyme in the ER that does this. It was all a hypothesis at some point. So what they were able to do is, well, first what people did is they looked at proteins that were translated in vitro. To do what's in vitro mean? I'm sorry, we're going way back here. 201 guys, what's in vitro mean?
Outside of the cell.
Outside of the cell, and it has to. If you want to translate a protein in vitro, what do you need? Okay. No, well, go. So, what are your big players in translation? Amino acids, RNA, ribosomes, right? And then tRNA, right? That's a big one too. Right. So you can do this. I can go buy a kit from Fisher Scientific, add in my RNA, and we can get protein. It's amazing. Amazing what we can do these days. So if you translate proteins in vitro, they're one size. But if you translate them with microsomes present, they're smaller. So the only difference here is you've got your in vitro translation system plus microsomes. And they're getting into those microsomes, but they're smaller. And so this was how people started to think about the signal sequence hypothesis. Well, maybe there's a signal sequence and it's being huge. And so they used these experiments to show that, yeah, something is going away when we add microsomes. Yeah. So in vitro, when you do translation, you get a protein, right? When you add the microsomes, they contain things that are in the ER, such as signal peptidase. So that's what I'm trying to get at. And then that signal sequence is going to be recognized and cleaved in the ER. And so the resulting protein is smaller because you cut off part of it. Make sense? Yep. Okay, so here's a practice problem where you guys can work in groups. You guys have already introduced yourselves to your neighbors. If you have new neighbors today, you can take a minute to reintroduce yourself after I go through this. And then I want you to spend a few minutes looking at this, thinking about this, because I like to ask questions like this on exams, and I want you guys to get used to the type of questions. So, you're studying IgG. an antibody secreted from B cells as part of the immune response. In vitro translation of IgG yields a protein that has a higher molecular weight than the protein that is secreted from a cell. Addition of microsomes to the in vitro translation experiment results in an IgG that had a lower molecular weight, identical to that of IgG secreted from B cells. Explain your results. So you do this experiment where it's just like what we talked about before, where you've got in vitro translated protein and then you have in vitro translated plus microsomes. Oops, just like right there. And then you run the gel. And then you do a Western for your protein. So this is a gel with a Western. M is for marker, that tells you your molecular weight. IgG, when secreted from the cells, about 100 kilodaltons. That's why it's lined up. This band here is lined up with 100. When you do the in vitro translation without the microsomes, you get this protein. So I want you to stop, think, talk to your neighbor, and we'll go over the answers or the explanation in just a minute. Okay? And I'll circulate, so if you have questions, call me over and be like, I have no idea what you're talking about. Because I love those questions. I'm so happy that you guys are so excited about the secretory pathway. So much excitement in this room. It's beautiful. Before I tell you guys, or before we talk about the answer, I just realized it might not occur to you that the molecular weight of a protein is dependent on the number of amino acids in it. Does anyone know that? Who knew that? Who didn't know that? Some people didn't know that. These are these things that you take for granted that people know, and you're going to have to stop me and ask me these things. So as a rule of thumb, you can move your decimal point. So if you have 100 amino acids, move your decimal point 1, it's about 10 kilodaltons. That's a little trick. So, so you can see how cutting off 10, 15 amino acids will affect the molecular weight of the protein, right? Because there's less. So it's going to be lighter. Okay, so someone just said, this is what you just talked about. Well, yes, it's just what I just talked about, but sometimes it's helpful to have you actually use your brain to work through it yourself. And hopefully everyone did. So who wants to take a stab at what's going on here? Yes?
In vitro just doesn't have the signal pathways to cleave off the signal sequence.
So what do you mean by in vitro? They're both in vitro, but what's the difference between the two samples? You're right. One doesn't have signal peptidase, but where is the signal peptidase? It's in the microsomes. Do you want to add to this?
No, I was going to say that since you don't have microsomes, you don't have the signal peptidase to leave off the extra.
Right, so if you don't have your microsomes, you don't have your signal peptidase. Remember, your microsomes are little ERs. What's happening with import of proteins into the ER? You have your signal sequence, it goes in, and then as the protein is translated in, then you have the signal peptidase that's hanging out right there by the translogon that cleaves off your signal sequence. And so this is exactly what's happening. Without your microsomes, you have the signal peptidase, you're going to have a longer protein. Does anyone need any clarification on that? So often I do these things. I think that's a nice break. I get to stop talking for a minute, and it's also nice for me to come and talk to you guys more individually, because sometimes people don't like to raise their hands and ask questions. And so you should always call me over and say, I'm lost. I have people say, I'm lost. And I'll say, well, when did that begin? And they'll say, the beginning. And I'm like, that's great that you're telling me that, because we can work on that. Okay.
Why
Yes.
did they use sucrose for the media?
Why did they use sucrose for the media? You know, people use different things to make gradients. Sucrose, I believe, is cheap. And really what you're doing is you're making different percentages. So it's so simple. You just weigh out your sucrose and do your 1%, 5%, 10%, all the way to like 25% usually. Yep.
What's the purpose of the detergent?
What is the purpose of detergent? What does detergent do, guys? Solubilizes membrane. Right. And what's your microsome made of? Right. Exactly. Exactly. Any other questions? Yeah. Why didn't they just use a what? So you're saying why didn't they do like gel filtration chromatography or something like that? They could have. They probably could have. This is requires less equipment, cheaper. You just need a tube and sucrose and a centrifuge. Whereas those gel filtration columns, you need the whole thing and the fractionator and all that. You need more stuff. And I think you need a really long column. And then I also question whether or not the membrane will stick, like if it's going to be sticky with the media. But that's good thinking. Good thinking. But I just got to think.
This would only apply to the secretory proteins, right?
This has to do with secretory proteins because they're the only ones that have the signal sequence being cleaved. So so someone said to me before, what does B cells have to do with this? And that's a distractor. You guys can ignore the fact that it's B cells. You just need to know that the protein's being secreted, so it's in the secretory pathway. And it could apply to any secreted protein. I could have changed it and said a membrane protein. I could have said an acid hydrolase, because those go to lysosome. Any protein in the secretory pathway could be substituted into this question. Now, if I did a nuclear protein, this wouldn't work because they don't go into the ER. Any other questions? Great questions we're getting. Keep them coming. Okay. Okay, membrane proteins. Integral membrane proteins are translated into the ER. Right, so we're talking about secreted proteins, membrane proteins, proteins that go to the lysosome. So these are membrane proteins that their final destination is the plasma membrane, the top of the surface of the cell, right? These are the guys, these receptors that are hanging out, binding to ligands and starting signaling cascades and all that stuff that you've learned about before. And so you have to ask yourself, how do these get here? Well, they're translated into the ER, and they move right into the membrane there. And then you might say, well, how does that get to the cell surface? Well, we're going to learn it's the membrane protein is in the ER membrane, and that is going to butt into a vesicle that's going to travel from the ER to the Golgi, to the plasma membrane. It's going to fuse, and that same membrane that's in the ER is going to become the membrane at the cell surface. Okay? So these proteins are going to move into the membrane co-translationally. You'll have that same exact thing that we just talked about, where you have SRP, binds to signal sequence, brings it over to the ER, then you're going to have translation into the translocon, and then you are going to hit this certain sequence, which is basically a big span of hydrophobic amino acids, and stop translation and move over into the membrane. Okay? So membrane proteins tend to have these transmembrane domains that are basically 20 to 30 amino acids with nonpolar side chains. Remember, amino acids have different properties. Who's taken biochem? 2 people. Who's going to take biochem? Okay, so you've learned it a little bit, you're learning it now. At some point you're going to have to probably memorize do you guys have to memorize all the amino acids of biochem? Now? Yes? Some people do. It's useful to know your amino acids, not to memorize them for the sake of memorizing them, but it helps you to understand proteins better and how they work. So for example, proteins that are very polar probably wouldn't be in a transmembrane domain, right? Because they want to interact with water. Membrane, fairly hydrophobic, right? So 20 to 30 amino acids is long enough to make a helix that's going to span the length of the membrane, plasma membrane. And then so it's usually in an alpha helix, and then the peptide bonds are more buried. The hydrogen bonds are buried inside the helix. Okay, so who's seen one of these before? Good, 2 people. Fantastic. This is a hydropathy index, which is basically a measure of the hydrophobicity of an amino acid. And so if it's above the 0, it's hydrophobic. If it's below the 0, it's hydrophilic. And so what happens is each amino acid in a whole protein is plotted based on how hydrophobic or hydrophilic it is. And you can start to identify transmembrane domains based on this. A transmembrane domain is the part of the protein that's likely to go through the membrane. Would you expect a transmembrane domain to be highly hydrophilic or highly hydrophobic? Hydrophobic, right. And so what we're showing here is this big peak here. So that starts at about amino acid 70 and goes to about 90. That's about 20 amino acids that are very hydrophobic. So you could say, oh, maybe this has a transmembrane domain. This is useful if you are studying a protein that has no known function. You can start by doing something like this, looking for transmembrane domains. There are these programs. You don't have to sit there and plot it out yourself. You just take the sequence, copy paste into a program on the internet, that thing on the computer, press the button, it's out there in the ether. And it will give you a plot like this, and then you can predict whether it has a transmembrane domain. This always confuses people, so I just want to take a minute to ask you guys, is this clear, or would you like some clarification? And if you'd like clarification, in what form? Are we good? Okay. It only takes 4 times of teaching this class to get these things out my mouth the right way. Okay, who's taken 410 or is going to take 410? I believe in 410 you guys are identifying, so Dr. Hem has this huge list of proteins, E. coli proteins with no known function, and you have to pick sequences that you want to try to clone to see if it's a real protein or not. And one of the things he has you do is this, because you're more likely to be a real protein if you have a transmembrane domain, I guess. I don't know. This is one of the gifts. You do like 10 different things. Yes? So how do you tell whether it has a transmembrane domain? So if you have 20 to 30 amino acids that are hydrophobic, you can guess it might be a transmembrane domain. So you do this. You see that this region here is about 20 amino acids. And you can say, okay, well, that's really hydrophobic. Maybe it's a transmembrane domain. Then you have to obviously do more experiments. You're going to have to, the good one is do, you can purify proteins, you can purify membrane fractions and see if it's there. You can do immunofluorescence, use an antibody to see if it's on the plasma membrane. There's a lot of things you can do to confirm where proteins are. Good question. Anything else? Okay, so integral membrane protein topology. I have topology bolded because I always get that question of what is topology. We kind of already brought it up when we were talking about microsomes, right? They have the same topology as the ER. The same stuff that's on the inside of the ER is on the inside of the microsome, such as signal peptidase, right? The ribosomes are still on the outside. The proteins are still translated onto the inside, right? So with integral membrane proteins, the topology is maintained all the way from the ER to the plasma membrane, meaning whatever is on the inside stays on the inside all the way through. And you might say, okay, what do I care? Well, what you're going to see is everything's going to be budding from the ER, fusing with the Golgi, budding from the Golgi, fusing with the plasma membrane. And it's really important to maintain the topology because a membrane protein, well, it does something, right? Can someone give me an example of what a membrane protein, like some membrane protein you can think of in your mind and what it might do, or what a protein that sits on the plasma membrane might do? Who's raising their hand? Oh. Acts as a receptor. So a lot of membrane proteins are receptors, exactly. And so on the outside of the membrane, what do you think that part of the protein might be doing on the outside of the plasma membrane? What would it be binding to? Ligands, right? Signaling molecule, some sort of protein secreted by some other cell, going to bind to that extracellular portion of your membrane protein, your receptor. And what's going to be happening with the inside of that protein, whatever's in the cytoplasmic side of the plasma membrane?
Sending the signal into the cell.
Sending the signal into the cell, so it's going to be interacting with other proteins, being phosphorylated perhaps, binding to other things. We're going to get there when we do signaling, but remember, this is all connected, okay? Right, so it's really important to maintain the topology all the way through because if it flips and you have your intracellular domain on the outside of the cell, is that going to be good for that signaling pathway? No, because suddenly you can't bind the ligand anymore and you can't signal. So it's important to maintain the topology. So, and the topology can be varied, right? So you can have your amino terminus on the cytosolic side, you can have your amino terminus on your ER lumen or your cell exterior, as you'll see it will become. You can have these multi-transmembrane paths receptors, which will have, you can have both your amino terminus and your carboxy terminus on one side or on the other side or one on each side. It's all specific to the protein. Every protein has its own structure, its own function. The goal though is having what needs to be there where it needs to be. Like the part that binds the ligand hopefully ends up outside the cell. Okay? Make sense? Eh. Hopefully. How are we doing here? Okay. Okay. So I'm saying that these membrane proteins are put into the membrane at the ER, right? So, for example, this is your simplest example, a single pass transmembrane protein. So it's got one transmembrane domain. One side of the protein is on the inside, the other side is on the outside. It's going to be integrated co-translationally into the ER membrane. And so it's all about this stop transfer sequence, right? So this is kind of what we talked about before, where you'll have a signal sequence, where in this case they're calling it a start transfer sequence, but it's a signal sequence. It's going to, just like all the other proteins we were talking about that would be in the lumen of the ER, it's going to bind to the translocon. You're going to get translation into the ER, but then it's going to hit the stop transfer sequence. And it's going to do exactly that, stop transfer. So it's going to stop, and it's going to move into the membrane. And so then you're going to get the rest of the protein translated, and that rest of the protein is going to be outside of the ER.
What happens to the signal sequence? Looks like a
Signal sequence always gets cleaved.
I'm
I'm going to take that back in this case. Huh?
Does it stay in the plasma membrane? Like,
You know what? So the question is, what happens to the signal sequence? Does it just stay in the membrane? Yeah, that would be silly, wouldn't it? If it just stayed there. All proteins have a half-life and end up being degraded at some point. My guess is over time they will be degraded somehow. I'm not exactly sure because you can imagine, well, like over the life of the cell, then you would have these membranes that are 100% protein. And that's not good. So they've got to go away somehow. But that question is, yeah, I don't know. I'm already getting people begging for credit over here. Okay, we'll do our first Stump the Teacher on the second day of class. Horrible. Did she plant that question?
No, sorry.
Okay, so this is a Stump the Teacher. So the question is, what happens to that signal sequence after it's cleaved? She's talking about this red thing here. Does it stay in the ER membrane, which then becomes that plasma membrane? Does it stay there forever? What happens to it? Now, you can tell me what you think, but you won't get extra credit. You have to have something fully referenced. It cannot be copied and pasted. If it looks like you went Google, copy, paste, send, you don't get extra credit. You actually have to think, and the answer has to go something like this. In class, we were talking about this. From my research, I found X, Y, and Z happens, and here's the reference. And you have 24 hours, so that means by tomorrow morning, you have to email it to me. You know what I need to do? I keep meaning to do it on Blackboard. Let's see if I can get my act together. For this one, email it to me, and then I'll set up something on Blackboard so you can do it through there. And if you are correct or even close, you'll get 1 point. Yeah.
What's the full question?
The full question is, what happens to the signal sequence after it is cleaved by signal peptidase? Does it stay there forever? Good question.
Can you repeat that?
It's recorded. It's all recorded. What happens to the signal sequence after it's cleaved? Okay. You guys just don't want me to finish my lecture today. Okay. Okay, so this is just another way to think about this. So you'll have your mRNA and your ribosome and your protein is being translated into the ER. Remember, here's your signal sequence. It was cleaved off. And then you'll get to this point where the stop transfer sequence, these 20 to 30 amino acids that form this hydrophobic helix, that is the transmembrane domain. That will show up in your translocon and it'll be recognized and translation will stop. It will move over into the ER membrane and then translation will continue on the cytoplasmic side to finish the protein. And so that's how you end up with luminal side, transmembrane domain, cytoplasmic side of a transmembrane protein. This protein will end up at the plasma membrane surface. You'll see, I promise. Yeah?
So does that mean that a protein can have multiple, like, transmembrane domains?
Yeah, and I think I have one coming up there. Yeah, the protein could have multiple transmembrane domains, and that's how you have, like, in G protein coupled receptors, they tend to have some, I think 7, I forget how many, but multiple transmembrane passes, and they would have multiple stop transfer sequences. So it would be like translate, stop, move over, translate, stop, move over. Yeah.
Is there like another single sequence to where you rest that protein in when they go multiple times to the membrane, or is
Is there enough? So you have this first signal sequence, and its major purpose is just to kind of bring it over to the ER and begin that translation. And then with every stop transfer sequence, you're going to move into the membrane, but then you'll still have, so you can see like here's your stop transfer sequence, you're going to move in, but then you're going to have the rest of the translation. But let's say there was another stop transfer sequence, right? So that would actually bind, it would actually bind in here and then move over again. I think I have a figure that shows that coming up. Other questions? So you can have an internal signal sequence. So what I just showed you was the simplest. We've got your signal sequence and then the stop transfer. Signal sequence brings everything over. Stop transfers your transmembrane domain and moves it into the membrane. You could also have an internal signal sequence, which actually acts as the same thing as a stop transfer sequence. And it's used to also integrate that single pass transmembrane protein into the ER membrane. And you can have it in two different ways. You can have it so your amino terminus is on a cytosolic side, or so your carboxy terminus is on the cytosolic side. It really depends on the protein, and it depends on the sequence surrounding that internal signal sequence. So in this case, the signal sequence is serving two purposes. One, to bring the protein over to the ER, and two, to integrate into the membrane. Okay? Questions? Yes? Yeah, so in this case, if this is, rather than having a signal sequence, that's going to, the purpose of it is to bring the protein over to the ER. Here you're having one sequence that's both bringing the protein over and integrating into the membrane. Yep. Okay? Yeah.
This is only with co-translational.
Yeah. This is all co-translational. I believe you would not have post-translational integration of proteins into the ER membrane. I guess you never know. I think we're done. Is that what people are telling me? Okay.